Okay, hello everyone. Thank you for having me. I'm here today to introduce the new replication initiative that we started called Revision, which is a replication initiative for image-related neuroscience. And just to back up a little bit, why are we even doing this initiative? So in visual neuroscience, our goal is sort of to gain an understanding of our representations of the visual world. So if you imagine this cloud on the right as everything in the visual world, every image you can imagine, then we want to gain an understanding of our representation of that, right? And a few years ago, people started using so-called condition-rich designs, which try to cover a broad part of this representational space of stimuli. So most prominently, we have things like Bolt 5000, things fMRI, or probably the most popular one, NSD, the natural scenes dataset. And these designs have some implicit assumptions that underlie them. And specifically, first, it's that they try to sample the image space broadly and sort of representatively. And secondly, that they yield generalizable findings so that the things that you find from them also hold for other images that maybe are not specifically sampled, but that are also part of this visual space that we're looking at. And our question that we wanted to ask is that actually the case? So do these assumptions hold? Do they sample the image space broadly and do they yield generalizable findings? And there are some findings that show that maybe this is not the case. So there was a study from last year by Shirakawa and Kamitani, and that study showed that for NSD specifically, but this likely holds for all of these data sets, that there's an undersampling of relevant areas of the semantic space, so that the image diversity is not actually that high and that there are quite a few areas of the semantic space that are still uncovered by these data sets. And then secondly, that there's also an oversampling of other semantic areas. So for example, if you have an image of a surfer in your training set, then likely there's going to be a similar image of a surfer in your test set as well. And that can lead to leakage and can lead to sort of an overestimation of your performance. And they showed this specifically for decoding or for image reconstruction studies that we may overestimate the performance in these data sets, but this may also be the case for any other study that uses trained test splits. And to address these limitations or to create a resource that maybe tries to address these biases, we created the LAION-fMRI dataset. This is a project, a sort of a large fMRI dataset of 5 subjects that all went through 30 sessions of 70 multi-echo fMRI sort of image viewing sessions. We also have a bunch of other sessions like retinotopy, diffusion imaging, all manner of other stuff. Participants viewed more than 25,000 images that broadly cover the image space that were presented 4 times, but we also have more than 800 images that were deeply sampled, so they were repeated 12 times. And lastly, this dataset also includes rich human metadata for all of these images. And this was an effort led by Josefine Zerbe, who's a PhD student in our lab. And now, how does this data set actually sample the visual world? So I said that we try to cover it broadly. This data set is based on LAION Natural, which is a data set of 120 million natural images that are subsampled from LAION 2B. And using these images, we try to really cover the semantic space as best as possible using just 25,000 images. And how that was done was that 10 million samples of this LAION Natural data sets were taken. Then we did a clustering to isolate some candidate images. And then always the image was chosen that maximizes the effective dimensionality of this data set. And this image was then added to the data set. And then the same process was repeated. So this was really meant to maximally have some sort of maximal effective dimensionality, which describes how many axes of variation are covered by a data set. And with this method, we actually managed to really have a very broad semantic coverage, also compared to other data sets that are out there. And this data set is now available. If you want, you can go to this QR code to find all the resources that you need to use this data set. And we also have a VSS talk recording from Josefine of her introducing this data set and sort of explaining a bit more of the noise ceilings that we managed to reach and all of these different things. Just one more thing, we also have a data loader that is actually really cool and makes it very simple to use this data. So you just have to install the data loader. And then just using two functions, so you just initialize the data set, you set a path where you want to store the data set, and then you can download the data set for all subjects. This is really all you have to do to get this data. But there are also more functions. You can also just download a specific session, for example, or you can download including all of the stimuli, something like that. Right, so this data set is now available. And to go back to the question that I led with, um, do these existing findings actually replicate on LAION-fMRI? That's what we want to find out. And secondly, do they also generalize to this broad image space that we're covering with LAION-fMRI? And because these are really big questions and we cannot answer them alone, we started the Revision Initiative, which is a replication initiative for image-related neuroscience. And before I explain more about what this is, I want to talk about why I think you should participate in this initiative. So there have been similar initiatives, most prominently the initiative published in Science, which estimated the reproducibility of psychological science that started the whole replication crisis in psychology. But there are also more recent examples of similar replication initiatives. And we also have replication initiatives in neuroscience. So the Many Labs initiative that is looking at EEG, but there's also other examples that use fMRI. And what all of these initiatives did is they recruited as many participants as they could. And then all of these participants replicated either a study or a finding. And then they sent a report to the organizers. And then the organizers wrote a big replication paper with all of these participants as co-authors on the paper. And we're planning to do the same thing. So if you participate, you also have the chance to be a co-author on our consortium paper that we're going to write in the end. Secondly, we also have prizes. So we're going to give out a replication award for the best replication attempt of $2,500. We also have the same for the best generalization attempt. I'm going to explain more what that means later. And then lastly, we have a facilitator award of $1,000 that is awarded to the most helpful original author. So if you're an original author of a paper and someone approaches you because they want to replicate your study and you're very helpful to them, maybe you help them with metadata or with implementing your code or whatever, then they can nominate you for this facilitator award. Now, one thing that is good to mention is that these prizes do not depend on whether the replication or generalization was successful. It's really just about how rigorously it's conducted and maybe how well written the report is in the end. Now, who can participate in this? Generally, everybody can participate. Overall, we want teams of 1 to 3 scientists that can participate together to replicate a specific study. And they can be from any academic level, really. So if you're API and you want to get back into sort of hands-on research, or if you're a postdoc or a PhD and you just want to be on a cool paper, or if you're a master's student and you're trying to sort of get into the field, get some experience with fMRI analysis, or you're still looking for a project for your master thesis, you can also participate. We also talked to people already that are going to structure a course around this at the university, which I think is really cool. So they're gonna have master students that in groups are gonna replicate papers, and then they have a PhD or a postdoc that is going to supervise them, and they together will build sort of this replication team that is replicating together. Participation, in our estimation, takes a few days if you're very skilled. And if you have a study that is simple to replicate, you can just change the data path in the code, run it with our data, and that's really it. But at most, it should take a few weeks. So even if you're new with fMRI data, it shouldn't take too long overall, depending on your experience. Now, what do I mean when I say replication? So the definition that we're going with here is that you replicate, or not the definition, but the goal is that you replicate the main findings of a paper using the same methods, but using the LAION-fMRI dataset. So you don't need to collect your own data. You can just use the LAION-fMRI dataset for this. And the main findings here, so you pick a study either from a list that is on our website, or you can also choose a study yourself as long as it's technically replicable using LAION-fMRI. And then you replicate the main findings that are mentioned in the abstract. So this is how we define the main findings. They should be mentioned in the abstract. And most of the time, there should also be a figure that is showing these results or these findings. Now, regarding the methods, we really want you to use the same methods as the original study, and if possible, also the same code. So most modern studies have the code online, and you can just use that. If you need help with that, you can always contact us, or even better, the original authors to get help there. And you should only diverge from these methods if necessary. So for example, because we only have 5 subjects in the LAION-fMRI dataset, you need to use single subject statistics. So if you have a study that uses 20 subjects, that just uses a between subjects t-test. You need to convert that to a single subject method. And the method that we chose is permutation testing, but we have a very detailed tutorial on how you can do that on our website as well. Now, for the generalization part, good to mention here is that this is not mandatory. So you can just participate by only replicating. But in many cases, doing the generalization will be quite easy. So the question here is, do the findings generalize to this broad image distribution that we have in LAION-fMRI without any overlap? And for that, we created a few different trained test splits that we implemented in LAION-fMRI. So if you imagine that this is the LAION-fMRI image distribution, the first trained test split that we created is the independent within distribution trained test split. And here what we try to do is really try to maximize global coverage of both train and test splits while minimizing the local overlap. And what that means is that both train and test split, they cover this broad space. So both will have images of food, images of animals, of cars, everything in there. But locally, they will be dissimilar from each other. So you will have animals in both, for example, but maybe you will have zebras only in the train set and then giraffes only in the test set, so that we don't have sort of exactly the same image in train and test set, just from a different perspective. Secondly, we have out of distribution clusters that we created. This is really just a simple k-means clustering of this image space. And this is meant to minimize the global overlap and the local overlap. So for example, you would have animals clustered out in its own cluster. You can train on everything else and then test on the animal images. And then lastly, and most stringently, we also have specific out of distribution images. So those include some more abstract images like Gabor patches or shapes, but we also have more natural images like natural illusions or sort of unusual relations, unusual images. And here you can really just train on the main image distribution and then test on the out of distribution images. Important is here that you don't just use all of them, but maybe you pick some specific groups that you think make sense to test this. And these three methods really give us three levels of testing if a finding generalizes, and three levels of stress testing. And as I said before, this is not mandatory, but for many studies, this will be very easy. It's just changing the way that you split the data. And of course, only if you do the generalization, you're eligible for the generalization award as well. And I explained it now for studies that use trained test sets, but you can also imagine other methods. So if you have representational similarity analysis, for example, you can also just test these different spaces independently. But we also have a more detailed description on our website about that. Right. So what kind of studies can you actually replicate? So of course, the most straightforward studies would be those that use a condition-rich data set like Bull 5000, NSD, or Things fMRI. But there are also many studies that use private data sets that can be replicated using this data set. And you can really replicate studies from all different areas of science. So you can use encoding models, cortical organization, image reconstruction, object scene processing, representational geometry, and probably many other areas, as long as the study is in principle replicable with LAION-fMRI. So this is important. But apart from that, you can really choose any study that you want. Now, most importantly, how can you actually participate in this? So it's pretty straightforward. What you need to do is you need to go to our website and then sign up to replicate a specific study, either from the list that we have there. So we have studies you can browse through, but I'm sure we left out many studies. You can also pick one yourself as long as you think it's replicable. And then after signing up, within a few weeks, you should write a proposal. This is really very short and simple. So it's really just, what is the study? What are the main findings that you isolated? And how would you replicate them? So this can be done very fast. We also have a template that you should use to write this proposal. And then you send it to us. And we have actually a board of reviewers, which are expert professors from the field, from like different parts of the field, that will review these proposals. This is just very general at this point. It's just they're going to look at it. They're going to see, do they think it's feasible? Does this make sense? Is this in principle valid and replicable? And then they mostly will just give you a thumbs up or a thumbs down, or maybe like, you need to adjust this. But in most cases, it will be very straightforward and they will tell you, you can go ahead. We also ask you that you send this proposal to the original authors. On the one hand, because we want to sort of keep them in the loop, let them know that we're replicating their study, but also because they can be very helpful. If you have any questions, they can help you with implementing their code, for example. Then you download the fMRI dataset, and the LAION-fMRI dataset, and you conduct the replication. And then at the end of it, you write a report, which is really just writing down everything that you did and the results. We also encourage you that you create the same plots that the original study created, just so that it's as comparable as possible. And then, of course, we also want to know your conclusion. But also for this, we have a template that you can use. And then you send this to the reviewers. We have one round of reviews here that some reports may get. So this is really just about, is this report valid? Is this replication valid? Is the conclusion valid, maybe? And then they will give you some short feedback. In many cases, they probably won't, and they're just going to say it's okay. But in some cases, they may tell you, maybe you adjust this, or you still look at this. And then you have some time to adjust it in the end. Now, to conclude, what is our timeline? So we started this initiative in May at VSS. And now you have time to write the proposals until the 15th of September, which is the deadline. But it might make sense to actually do that sooner, because the sooner that you send us your proposal, the sooner you're going to get feedback on it, and the sooner you can start replicating. And also, if you have a specific study in mind that you want to replicate, it might make sense to be fast, because if you sign up for a study, it will be blocked for other people, right? So this is also why you should not wait too long to write your proposal afterwards. And if you have a specific study, you don't want anyone else to take it away from you, essentially. Right, then the deadline for the report is going to be the 15th of February next year. Then you're going to get feedback on that, if feedback is necessary, by the 31st of March. And then the 30th of April will be sort of the final report deadline, where you have then time to adjust some things. And then afterwards, we plan to write the revision consortium paper that you all can be co-authors on if you participate in this. Right, so I hope that you will join the revision initiative. We have a website, as I mentioned before, where we have all the information. So you find information about the image data set that we used, information about the fMRI data collection, but also information about the initiative, how you can participate, what you can get out of it, and all of these things. And if you click on participate, you can just fill out a simple Google Sheet, and then you're signed up for this initiative. So I hope that I could convince you. I would like to thank this amazing team of researchers that have worked so hard already to make all of this happen. And I hope that you will consider joining this team. Thank you very much.